One thing, a quick announcement. Next week's seminar, which is Thursday, right before some of us go over to New Orleans for the aquaculture conference, speaker is going to be Dave Bushick, who, among other things, runs the oyster aquaculture program up at Rutgers. So if you're into oyster aquaculture in any way, I hope you'll come by and meet him. There's some really good things going on up there with respect to oyster aquaculture. So I'm pleased to have, if I can undo my little thing here. There we go. Pleased to have Jane Caffrey today from University of Western Florida, or West Florida?
West Florida.
West Florida. Jane has got quite an interesting history. A master's from LSU, PhD from University of Maryland. She got a Fulbright scholarship, postdoc at USGS, and then a research position with USGS, became a research scientist at the University of California, and finally came over to Pensacola. So she's had an interesting history. And today she's going to talk about biogeochemical cycling in the Gulf of Mexico.
Well, thank you so much for the invitation. I'm really glad to come and talk a little bit about some of the work I've done in Pensacola Bay over the last almost 20 years. I've been in Pensacola 20 years, but not necessarily been researching Pensacola Bay that long. So I want to provide a little bit of historical perspective about Pensacola Bay. Some of you may be familiar with the area, our beautiful white sand beaches, clear waters, a little bit different than Mississippi Sound. But I think there will likely be things that you see that may be similar to environments in this location. So in some ways, Pensacola Bay is typical of estuary in the Gulf of Mexico. So I'm going to start out talking a little bit generally about eutrophication and nutrient cycling. That's often an issue in many estuarine environments, and Pensacola Bay definitely has some issues related to eutrophication. And by that, talk a little bit about some of the historical data and trying to uncover what has gone on in the past. And then transition into some of the research that I've done looking at biogeochemistry in the estuary, focused primarily on sediments, both in the hypoxic channel, in shallow sediments, and then habitats like seagrass beds and hard structures like oysters and artificial reefs. So I'm not going to go into anything in any great detail, but try to provide a bit of an overview. So the general way that we think about eutrophication is a result of human activities. The fact that as we increase nutrient loading to the environment, that leads to an increase in the organic loading. And so this is the Nixon definition of eutrophication. And most often we see this enhancement in phytoplankton production. Sometimes we'll see it as macroalgae. And there are a variety of things that will occur as a result of that increased organic loading, and some or a variety of those can occur in the systems. So we can have changes in the primary producers, whether it's phytoplankton, macroalgae, or seagrasses. We can have changes in the community structure of benthic invertebrates. One of the most serious things that often results as a result of eutrophication is increase in oxygen demand by the microbial community. And because of that increasing oxygen demand, we end up reducing oxygen levels to levels that are unsustainable for many invertebrate and fish communities. So I'm going to focus initially looking at sort of what we think of historically, what we know about Pensacola Bay, and then talk about what might be some of the impacts of hypoxia in the Pensacola Bay Channel sediments. So in estuaries, again, probably most of you are very familiar with this. This is perhaps a little bit simplistic of a diagram, but the physical factors can play a very important role in determining how serious a problem eutrophication is. So we have our freshwater inputs, often with high levels of nitrogen and phosphorus, and that can lead to enhancement of the phytoplankton community. And if we have sufficient fresh water coming in to cause significant stratification, that again can enhance bloom conditions because the phytoplankton are up near the surface where they're getting lots of light. And then as they sink down below the bottom, they get consumed through bacterial decomposition. And again, because of this stratification, we have less mixing between surface and bottom waters. We can get this oxygen depletion and hypoxia. So that can be a real serious problem in water columns. Even as shallow as many of our Gulf Coast estuaries, it can still be a significant issue. So again, probably most of you are very familiar with this. Of course, the Louisiana shelf is lots and lots of study has been done looking at what happens as a result of flow down the Mississippi River. So all of the agricultural areas in the breadbasket of the U.S. coming together, coming out the Mississippi River, coming out the mouth of the Mississippi River, leading to this extensive hypoxic zone. Lots of work's been done on that. I know that there are issues periodically in Mississippi Sound as a result of hypoxia. Chesapeake Bay is another very well-studied system with issues related to hypoxia. The Baltic Sea could do lots and lots of lists. Well, Pensacola Bay fits in this category as well. So Pensacola Bay, located in the western part of Florida, and again, to give you a sense of what the system is like, what the drivers are for this system. So we're located right here at the border between right next to Alabama. The watershed goes up into Alabama, so it has two states feeding into the watershed. It's generally a pretty forested area. We have some amount of agricultural impacts in some parts of the watershed, primarily things like peanuts and soybeans and cotton are some of the primary crops that are grown in the watershed. Trees are also a very important crop that are grown, lots of logging. And we have our urban development and suburban development. So in the Bay system, we have the city of Pensacola, we have the Gulf Breeze Peninsula, we have Pensacola Beach. And so this is where we have our major centers of population. This is where we have our most intensive increases and changes in land use compared to much of the upper watershed that still has some amount of preserved riverine riparian zones that are still fairly intact. So if we kind of zoom in a little bit closer into the Pensacola Bay Estuary, these are some of the characteristics. So major freshwater flow coming into the Escambia River. About 70% of the fresh water comes in to this part of the bay, the Escambia River, Escambia Bay part. We also have significant fresh water coming in from the Yellow and the Blackwater systems. Minor contributions from kind of more local streams. The city of Pensacola here, we have some urban bayous. And so this color contour shows that we have a lot of area that has very shallow shoal regions. We have a channel that is maintained. And again, like most of the Gulf of Mexico, a microtidal sort of system. And so I'm going to be talking about some work done at a variety of different locations. Early on, when I started working in Pensacola Bay, very heavily focused in the channel, looking at what's happening in this zone of the bay and the adjacent shallow areas. And in more recent years, I've been doing more work in Santa Rosa Sound and Big Lagoon, which are both seagrass systems. And so we'll look at what's happening there. Other things that may be sort of interesting about this, again, fairly shallow in depth, about 3 meters average water depth. a residence time of about a month in terms of the water residence time. So again, like many systems, it has changed over time. It's responded to human inputs in the estuary, and like many places, it suffers from a variety of contaminant issues. There was a major PCB spill in 1969, and we're still able to detect PCBs in some parts of the bay downstream of where that spill occurred in sediments, and also in some of the fish species that are resident in those areas. We have a coal-fired power plant. They do have natural gas. They do have the more advanced scrubbers that they've added in. But nonetheless, they do use coal for some amount of power generation. We have a paper mill, and so also attendant issues, concerns about contamination arising from producing paper. They also will use some amount of coal for their industrial boilers. So again, all the concerns about having coal emissions and what effect that has on the environment. We have a variety of Superfund sites within the region, and they have done cleanup activities. Pensacola Bay, the Pensacola City area has one of the unfortunate distinctions of being one of the places where they had to relocate citizens because the contamination was so extreme that it was not safe for people to stay in their houses because of the contamination from one of the Superfund sites. And then, not surprisingly, again, like many coastal areas, it's a very desirable place to live, very beautiful waters, everybody wants to come live there. So we have this increasing coastal development, and associated with that, people often want to have their perfectly manicured lawns right up to the water's edge. So we can definitely see a wide variety of impacts that humans can have in the Pensacola Bay system. So if we try to tease apart what happened historically, it's a little bit of a detective story to try to figure out what's going on, because very often we didn't really keep very good records of the kinds of things we might be interested in now. So spent a little bit of time trying to understand how conditions have changed over the last 50 to 100 years. So the USGS has been monitoring freshwater flow in the Escambia River just below the border with Alabama since the 1930s. And if we look at the long-term annual trend in discharge, we have wet years, we have dry years. Maybe it's a little bit more variable. I haven't done a trend analysis. I don't think there's a trend in changes in freshwater flow that we see at this point in time. The USGS has also done extensive work looking at fertilizer use in all of the counties, and so drawing on that data, looking at nitrogen and phosphorus inputs from fertilizer. So this USGS data is, again, probably quite similar to many other locations where we see a rapid increase in use of nitrogen fertilizers following the Second World War, and then perhaps a pretty nice decline here, perhaps due to changing use of crops. Again, haven't really gone into it in great detail, but trying to understand what the overall patterns are. Some changes in phosphorus inputs over time. And then the biggest impact clearly is the change in populations in the Pensacola Bay watershed area, and so generally very low population density, and really not kind of slow growth. And then following World War II, we see this very rapid population increase. Again, similar to many other places that people have looked at this in Florida. Florida has became a very desirable place to live, and so we have this really big increase in population growth. So how might conditions have changed as we look at sort of the the nutrient inputs to the system. Again, drawing on data from the USGS, at the gauge site, in the upper river where they measure flow, they also have nitrate data. And this kind of smooth line is the lowest line. So this isn't the individual points, but it's the modeled sort of best fit of the points for nitrate concentrations. So since the 1950s, when they first started measuring nitrate in the river, we've seen about a doubling of nitrate. So we've gone from perhaps 7 to about 14. So those of you who've worked in very eutrophic systems, you'll realize that 14 is not really that high. Certainly not anywhere near as high as something like the Mississippi River, where it can be 100, 120 micromolar. But still, you know, this is a change in the system. If we look at what's happening in the bay itself, again, a little bit of a puzzle trying to go back, look for historic data, look for historic records. And so this is the mesohaline part of the Pensacola Bay system, you know, where salinities generally are in the, you know, 5 to 15 range, so downstream of where the river comes in. And so we have some data for nitrate in this region. Chlorophyll A, which is a measure of the phytoplankton biomass, they didn't really start measuring that until the 1970s, so we don't really know what it was in that earlier period of time, but we do have some amount of data. These numbers above represent the number of observations that went into the box plot here, so we can see the median and the 25th and 75th and the 90th and the 10th percentiles associated with the data. So we do see some very high nitrate concentrations in the 1960s. So this is before the Clean Water Act, as they were sampling in different locations. They were sampling near an outfall. They were getting some exceedingly high concentrations of nitrate in some of those areas. Again, you know, at these levels that we think are quite high. But if we look at kind of the recent data, you know, perhaps there's an increase. I'm not sure I would you know, put any money on whether this is, you know, that big of a change in nitrate concentrations. Chlorophyll concentrations, we do periodically have some high chlorophyll concentrations, as much as 20 micrograms per liter, which is that's a decent sized bloom of phytoplankton in the estuary. But in general, chlorophyll A concentrations are, you know, in the middle part of the bay are generally around around 7 or 8 micrograms per liter. So not really any huge changes that we can see in this system over this 40-year period, where we have some amount of data, some reasonable understanding of what's going on in the system. What we do have is a historic loss of seagrasses, and there is some amount of evidence to suggest that a lot of the shallow areas in Pensacola Bay had very extensive seagrass beds. Again, a little bit of a challenge to reconstruct it because we didn't actually have people going out and surveying it. So it's based somewhat on aerial photography and people's recollections and some sort of written documentation, but not as much quantitative information as we would like. But based on examining aerial imagery, we do see that there have been significant declines in the seagrass beds in the Pensacola Bay system. We generally have the seagrasses that we find in many of the marine areas: Thalassia testudinum, turtle grass, Haliduli raitai, Cuban shoal grass. We also have Rupia maritima, which is quite extensive. In some of the freshwater areas, we have the freshwater species like Vallisneria americana. We do have a bit of Syringodium, not as much as many other places in the Gulf, like in Texas. But in general, we recognize that we have had significant losses of seagrasses in the Pensacola Bay system. And some of it may be nutrient related, some of it may be related to turbidity in the system as a result of land use practices. We have highly erodible soils, so we can have sediments coming in. Those sediments coming in can shade out the seagrasses and contribute to losses, increasing coastal zone development. Despite the fact that we have significant losses, there are areas where we have very healthy seagrass beds. So areas like Santa Rosa Sound and Big Lagoon, we still have healthy, pretty thriving seagrass beds. So we definitely have this change in seagrasses that, you know, human activities have probably played a significant role in. We also have hypoxia in the Pensacola Bay system. And this is some work done by Jim Hagee and Mike Merle, working out at the EPA lab, lent me their slide on this. And so they had a series of cruises that they did down the Pensacola Bay system from the freshwater to the marine end. And we can look at how oxygen levels change as a result of river flow. So during periods of relatively low river flow, we have pretty good mixing. We don't have much in the way of stratification. Oxygen levels are pretty good. During average river flow, we can have much stronger stratification. So with the fresh water on the surface, inhibiting mixing of the bottom waters up to the surface, higher plankton production, we can have the development of hypoxia in the bottom waters. We really hardly ever get anoxia. We definitely get down to 2 and maybe a bit less than 2, but I never think we've ever really gotten down to 0. And then with very high river flows, it can be so fresh that the upper part of the estuary is essentially fresh, and this hypoxic lens can be displaced further down bay. So if we look at this sort of mesohaline region and sort of what the conditions are there, we can see some kind of interesting things going on both in the water column and in the sediments. So that's kind of the spatial pattern. We also spent some time, Mike Merle and I spent some time looking at dissolved oxygen records, and again a box plot kind of showing the seasonal pattern in oxygen conditions in the Pensacola Bay system. So again, the number of records that went into the box plot, the median, and so while it is possible to get hypoxia outside of the summer months, it really is most likely that you're going to see it during those summer months, July and August, and that really makes sense. Higher microbial activity, greater consumption of oxygen, much more likely to get hypoxia, but it still is possible to get it, you know, kind of outside of that zone as early as May or even into October. So what effect does these low oxygen concentrations have on nitrogen and phosphorus cycling in the system? So if we look at the concentration of nutrients in the bottom water in relation to oxygen, that's what this plot shows here. So we have our oxygen concentration on the x-axis and either our ammonium concentration, which is the filled in dots, or our phosphorus, dissolved inorganic phosphate concentration in the open dots. We can see that as oxygen concentrations get lower, we get this increase in ammonium concentrations in the bottom water. And this is not particularly a surprise. This is something that is often documented in hypoxic systems. It's primarily a result of loss of nitrification, which is a process I'll talk about in a little bit more detail. But that ammonium stays in the form of ammonium. It doesn't get oxidized to nitrate under these low oxygen conditions. For phosphate, we don't really see a big spike or increase in phosphate, and that's a little bit different than some other systems. Some of the places like the Baltic and I think also the Chesapeake Bay, when you get down near 0, you can have a release of phosphate. increase in phosphate concentrations. We haven't really seen that in the Pensacola Bay system. If we look at what happens to the fluxes, so if we were to take sediment cores and incubate them and measure what the ammonium and phosphate release is from those sediments over time, we can look at how oxygen relates to those measurements. And again, we see kind of a similar pattern where as we reduce oxygen concentrations, we get this increase in ammonium flux coming out of the sediments. But for the phosphate fluxes, we don't see any change there. And again, I think it's this is maybe something characteristic of the system, or maybe a little bit of the fact that if we don't incubate them low enough, we don't see it. Sometimes people haven't really seen it until oxygen gets really close to zero, and I think the lowest we got with any of our incubations was down around 1 milligram per liter oxygen. So it's possible if we were to do experiments where we basically removed all the oxygen, we might see phosphorus coming out, but based on the measurements more in line with what the field conditions were, we don't see any response to the phosphate with the change in oxygen. So just to kind of give you a visual of what the sediments look like in the Pensacola Bay system. So these are typical sediments from the channel, very fine grain, some amount of benthic invertebrates, kind of the ones that are tolerant of hypoxia. So we do have some of the polychaetes that we will find in there. Generally not much of an oxic zone on the surface. The shoal sediments from the shallow areas, generally much sandier, generally have a very low iron content to them, and often have this very oxidized layer. And one of the things that is probably most different between the Pensacola Bay system and your Mississippi Sound system is that the Pensacola Bay system is a very high light environment. So there is a significant amount of light that reaches the bottom. We estimate that we can have significant benthic production occurring within these shallow sediments, and even out into some of the somewhat deeper areas, because it's generally a pretty clear environment. So we've done a bunch of work looking at benthic fluxes in the Pensacola Bay system. I've also done some work with students looking at nutrient fluxes in seagrass beds. For those experiments, we were using benthic chambers. So we were putting chambers down over the sediments and measuring changes in nutrients and oxygen concentration. So I'll show you some of those comparisons. And we've also done some work looking at microbial rates. I was doing some work again in these channel and shoal sediments. And I had a graduate student who was looking at nitrogen fixation, and I'll just very briefly show some of that work. So one of the big takeaways from this looking at the relationship of light and benthic production is that even in some of the deeper sites, you can still have enough light getting to the bottom so that you can have gross primary production in some of the deeper sites. So these were some experiments that the EPA folks did that I was able to piggyback and do some additional sampling alongside them. And so, whoops. So basically, the greater the amount of light that reaches the sediment surface, the higher the benthic primary production is going to be. And so these, again, were all these core incubations that they did. So these are all the real shallow water sediments, the shoal sites. These are the channel sites, and even at some of these channel sites, there's enough light that reaches the bottom that you can get benthic production. So if we kind of look at the oxygen balance and compare some of these different locations, and see what kind of differences we can see in the oxygen and the nutrient fluxes. So these box plots are color coded. So the green ones are from the seagrass beds. So these are the ones that were done with the in situ chambers. The red are the channel sites, and they were done with core incubations. And the blue are the shoal sites, and they were also done with core incubations. And most of the channel and the shoal work we were doing in the summer and a little bit into the fall. So, not surprising, we have high production in the seagrass beds. Not much difference between the channel and the shoal sediments in terms of the net oxygen production. A little bit higher in the shoals than in the channel, particularly in the fall. If we look at respiration rates, again, the seagrass beds, highly organic, very active microbial community. Very high respiration rates going on in there, and again some lower rates in the channel and the shoal compared to the seagrass beds. If we look at the nutrient fluxes for these sites, if we look at the ammonium fluxes, we can see generally we have ammonium release most of the time. We have some uptake of ammonium in the seagrass beds, and some uptake in the shoals during the fall, but generally we have ammonium release, and much higher ammonium release from the channel sites. And so this is basically the different version of the same data that I showed. Those hypoxic locations were having more ammonium release. If we look at phosphate, generally low phosphate fluxes, some greater fluxes in the seagrass beds compared to the shoal in the spring, but not so much in the summer. Somewhat higher phosphate fluxes in the fall here, and then very low nitrate fluxes. So since this is a low nitrate system, we're not really getting a lot of nitrate uptake. We are actually getting some nitrate production. And so we do see some evidence of nitrification going on, the conversion of ammonium to nitrate at some of these locations. So if we look at the relationship between respiration rates and nitrate plus ammonium fluxes, the DIN flux for the different sites. This line here, it represents the Redfield ratio. So if if all of the material respired were to come out as inorganic nitrogen flux, it should fall along that line. And we do see that for some of the measurements in the channel, but we don't really see that happening in the shoal or in the seagrass bed. And this is primarily because of the high nitrogen demand by the seagrasses. So even though they're respiring material, any of that nitrogen that gets released as a result of decomposition, the seagrasses are taking it up, or in the case of the shoal sediments, it's the benthic microalgae that are taking it up. So they're not letting any of that nitrogen get out into the bay, they're incorporating it into their biomass. So we see this effect of demand of nitrogen by these communities. So might not really go through this in great detail, but the main point of the table is that we have these really high respiration. We have some very high rates of respiration and a little bit of ammonium in some of the Pensacola Bay sediments, kind of comparable, but a little bit lower to many other estuaries. And some of these estuaries where we have significant nutrient issues, we can see very high ammonium fluxes in places like Chesapeake Bay, very high phosphate fluxes, inorganic phosphate fluxes in some of these locations. Mobile Bay, we have very high phosphate fluxes. That's not the case in the Pensacola Bay system. So numbers are sort of similar, but in general much lower, and I think that's a large response to the primary producer communities. So this is just sort of what happens in a net sense when you're looking at a flux rate. I also was very interested in looking at the activity of a particular group of nitrogen cycling organisms, the nitrophiers, and this group will take ammonium and oxidize it to nitrite and then nitrate. So I've done a bunch of measurements looking at their activity using kind of optimal conditions to promote their growth. So adding ammonium so they're not short of ammonium, and doing them under fully oxic conditions, so if they need oxygen, it's present. So it's not supposed to be an in situ measurement, it's supposed to be what are the potential of these organisms under kind of optimal conditions, because they do require molecular oxygen in order to nitrify. So we do see that we do have high rates of potential nitrification occurring in the channel sediments in both the summer and the fall, quite low in the seagrass beds, high and somewhat variable in the shoals in the summer. And if we look at is there a relationship to oxygen concentration, what we see is that there is a microbial community there that is ready to be active as soon as conditions are right. So while we have that ammonium building up suggesting that nitrification isn't occurring, if they do get oxygen, the fact that they are have these high potential rates at low so this is the environmental condition. So when I collected the sample, this was what the condition was. If you give them oxygen, they will nitrify. So that means they're ready, they're primed, they're ready to be active when conditions are good. And so again, looking at what's happening in different environments, we do have generally low rates in the Pensacola Bay system compared to some of these other systems, but not wildly different. And again, probably in the seagrass beds, the seagrasses are much better at competing for ammonium than the nitrophiers are. They're just not able to be as good as the seagrasses are at taking up that ammonium. So the seagrasses are kind of winning out. And I did a bit of work with Tim Hollabaugh and others looking at what kind of organisms are doing this nitrification. So this is work from Tim Hollabaugh's lab, looking at whether they're ammonium oxidizing bacteria or ammonium oxidizing archaea. And in general, we find that it's primarily archaea. And if you look at the relative abundance of the archaea and the potential nitrification rate, we do see a positive relationship both for the channel sediments and the shoal sediments, although they are at two different levels. And this is again probably due to the relative availability of ammonium. There's a lot more ammonium in the pore waters of the channel, much higher pore water ammonium concentrations than you find in the shoal sediments. They're about 10%. So that's just not enough to keep the community as active as it is in the channel. So anytime we have that hypoxia goes away, the nitrophiers are primed and ready to be active. So this is some of the older work that I did in the Pensacola Bay Sediments, and so just to kind of give a brief summary of that work before moving on to some of the more recent work. Pensacola Bay, we see similar things to many other estuaries. We see a response to these human activities. So we do see things like hypoxia and loss of seagrasses. We do see this significant benthic primary production both by the benthic microalgae, Microphytobenthos, and the seagrasses, recycling, and then the generally low nutrient fluxes. So in recent years, I've gone back to doing more work in seagrasses and also looking at invertebrate communities, both oyster reefs and artificial reefs. So I'll very briefly touch on some of that work. So a little bit of work. One study that I was able to do on oysters, I think provides some interesting insights into the role of the microbial communities on oyster reef communities, and is likely generalizable to other invertebrate communities as well. So what everybody knows about oysters and is often one of the reasons why people are so interested in doing oyster reef restoration is because of their role in clearing the water and grazing down phytoplankton, and then also the significance that they have as a hard bottom habitat and refuge for many species as well. My interest has been looking at the microbial communities, and there's been a lot of work done looking at the effect of the pseudofeces or feces from the oysters and how that impacts nutrient cycling. But I've actually been interested in the oysters themselves. Are they sites for biogeochemical cycling? And so we did a set of experiments. Again, my colleague Tim Hollabau and Behzad Mortazavi, we did a set of experiments looking at nitrification rates, and we also did some denitrification work, looking at living oysters and then oyster shells, so newly dead oysters, looking at the activity on those shells. And again, doing these kinds of potential nitrification rates, we can see that we have nitrification occurring associated with the oysters on a variety of experiments. There's one that we did in October where they weren't active, but most all the others they were quite active, and a little bit more in the living oysters, but still even on the shell we can still see nitrification occurring. So these microbes, whether they're bacteria or archaea, living on the shells, nitrifying on using this substrate as a habitat. So from that work, I kind of pivoted and looked at some of this related to artificial reef structures. And so I had a graduate student who was looking at what happens as artificial reefs get colonized by benthic invertebrates. And so she put a set of limestone rocks out on the artificial reefs after they were deployed offshore, and would do experiments looking at the fluxes on those rocks. She'd bring them back into the lab and do experiments in the lab looking at oxygen and nutrient fluxes. And I also had a undergraduate do something very similar, using biofilm plates, and got very similar results to the limestone rocks. So having the limestone rocks is great, but you can see it basically on any surface as you get the community attaching and doing things. So if you look at the rates of oxygen, so this is oxygen consumption, and this is grams of ash-free dry weight. So this is based on the biomass of the invertebrate community and the microbial community. We actually added both of them in there. As you get more organisms on there, more biomass on there, you get greater oxygen consumption. You'd sort of expect that. Similarly, we see an increase in the ammonium flux, and again, you would expect that. These invertebrates are going to be excreting, they're going to be releasing ammonium. But one of the things that really excited me was the fact that we could see that as we got these more well-developed invertebrate communities, as these rocks had been out at the site for a longer period of time, we also started to develop a nitrifying community, because there's no other way to get production of nitrate than by nitrification. So there must have been nitrification going on within these organisms. So we can look at these relationships and see that, like we saw with the oysters, we can see this microbial community. And one of my colleagues at UWF has done has looked at the relative amounts of ammonium oxidizers and ammonium oxidizing both the bacteria and the archaea, and found evidence of both of these on some of the invertebrates. We haven't done a real in-depth study with it, but we do know that there are both archaea and bacteria on these artificial reef invertebrates. And so I'm going to pivot a little bit here away from this more research-heavy biogeochemistry into kind of the most recent kinds of things that I've been doing, which has been trying to do more work with citizen scientists. And so I have a colleague who is president of the Brim Fishermen Association, and they have done water quality sampling in the Pensacola Bay system since the 1970s, the early 1970s. And so they're always trying to get more people involved in water quality to understand why water quality is important. And so she has developed a partnership with the Pensacola Bay Oyster Company, and they're also interested in kind of having some community outreach as well. And so they developed a project where citizens would get baby oysters. The oyster company, would produce the spat, produce the baby oysters, and they would put them in these cages, and citizens would put them off their docks, or if they didn't have a dock, they'd match them up with someone who was willing to let the volunteer go out and take care of the oysters. So they would put them out as baby oysters and then monitor their growth. Barbara and volunteers would go out and measure water quality and kind of see how they were doing in different parts of the bay. So this was done throughout the Pensacola and Perdido Bay systems, in all the areas where Barbara thought it was most likely that they might get good oyster survival. So she recruited a lot of volunteers to help build the cages and to do all the sorting and to get everything all set up, and people who were willing to be a part of it. And so I thought this was a great opportunity to try to help provide a little bit more insight into what they were doing. So I've been working with them and the data that they've been collecting to try to understand how water quality will affect the growth of oysters. And I've also just started this semester trying to look at some short-term variability in water quality. They only go out about every 2 months to the sites, and so I have some sensors that I've put out. We're trying to look at more short-term variability to try to see what's going on. So this is just some real preliminary data from last year, from May and June. And so these are a variety of different places. So we have some urban bayous like Bayou Grande. We have marine areas like Big Lagoon, East Bay, Escambia Bay, which is kind of a more freshwater area, the lower Pensacola Bay system, over in Perdido Bay, the lower Perdido Bay system, Santa Rosa Sound, again, high salinity. So we can see that the oysters definitely grew between May and July. In some places they grew a lot more. East Bay they didn't grow very much. Escambia Bay they didn't grow very much compared to some of these other locations. So we have new data. They have data from October and data from January. We're still trying to crunch that. So we're going to try to relate that to the water quality that they're measuring at the same time that they're taking these measurements of the size of the oysters. So I think this has been a really interesting project and a very nice partnership. I've also, as I mentioned, started to get a little bit more back into doing seagrass work, and my master's student Rachel Capps came from UT. She was actually an undergraduate who worked for Kelly before when Kelly was working on her PhD. So she came very well trained. So I would take any students from Kelly that you want to send my way. I'll be happy to have them. So anyway, Rachel's study, she was very interested in nitrogen fixation, so she was looking at rates in both the Thalassia testudinum beds, Haliduli reitei, in nearby bare sediments, and trying to understand the environmental conditions that might have promoted this. I had an undergraduate who did a summer project and a little bit more looking at pore water biogeochemistry, comparing several different species, ammonium, phosphate, sulfide, iron concentrations in pore waters, and how that related to what kind of species it was, whether it was the freshwater species or the marine species, and how and biomass. So that's been interesting. And then another kind of citizen science monitoring project. So in 2016, I was working with FWC on a project to try to understand how what the roadblocks were to recovery of seagrasses. We know seagrasses are gone. What do we need to know to figure out how are they going to come back? So we did a lot of work. I didn't really do much of it. I had a really great crew that did most of the work. But we did a lot of surveying, and it really got me thinking that, you know, maybe would be a good thing if we could just have eyes on the ground on a regular basis. So in 2017, I recruited our Sea Grant Extension folks to help me find citizen scientists. And we started this citizen science seagrass monitoring program. It goes from May to October, the main part of the growing season, time when it's nice to get in the water. Not asking citizen scientists to get in the water in January when it's cold, not that useful. So they get a quadrat, they get a data sheet, and they get a water bottle. So they have been trained to identify the seagrass species. They estimate the percent cover in their quadrat. They try to take some additional measurements, write down any kind of notes they can about the sites, and then the water bottle, we get back to the university, and I have my students analyze it for total suspended solids and salinity, which are fairly stable, not really going to change that much if it's not analyzed right away. And then I try to bring my students out and we do more intensive field sampling, more detailed sampling with the YSI, and looking at nutrient and chlorophyll concentrations in there. So I think this has been a very interesting and very kind of fulfilling pivot from some of the things that I've done in the past, and I think there are real benefits to trying to combine students with citizen scientists. So I think for the citizens, they can be very passionate about the environment, but it's a way to bring information. It's a way to bring the science to them, so they can be more invested in it. They can be more aware of what's going on, what you can say, what you can't say. They can be much more empowered to know exactly what's going on at their locations because they're there. They're getting feedback about what we're finding, and I think it's a real benefit for the students. So they get real world application. They get to see why we do this. And it's also, I think, really helpful because, as they get a chance to talk to people, they get a way to really understand how to communicate what they're learning to other people as well. And it's often much more effective, I found, if I actually have my students talk to local citizens than if I do, because they can actually relate. They might have kids, they might have grandkids, and it really makes a big difference. So that's what I've been up to. So lots and lots of students. I would have pages of students because I have some students for a semester. I have some students for a couple of years, and lots of this work would not get done without all of them. And I'll take questions.
Questions?
So how old is that picture of Jim Hagee? Because I can't imagine when he's been in the lab last.
So that was the benthic pelagic study. So that was done in 2003, 2004.
Okay.
So yeah, so that's 15 years ago now. Yeah. Yeah. Yes.
So I was excited to see that Pensacola Bay is getting a new NEP.
Yes,
Based
yes.
on your work and then your understanding of that system, what are their biggest challenges that they can actually do something about?
So the biggest challenge is if the powers that be will actually do the kinds of things that we already know. And you know, it's so how important are private property rights versus the rest of the community? So if you have a piece of property and you want to put up a bulkhead, that's going to have an impact on seagrasses, it's going to have an impact on a whole lot of things. You know, you're fertilizing your lawn. Are you going to live in a flood-prone area? Are we going to allow the developers to put homes in areas that are likely going to flood? Are we going to allow DOT to build roads that we know are going to get washed out? You know, it's, you know, for so many of the issues, we actually don't need more science. We really just need people to apply what we already know. And you know, and that's why I'm trying so hard to work with citizens. Because I feel like if I can get enough citizens with eyes on the ground that are interested and empowered, and that they are going to the county commissioners saying, no, we don't want you to dredge. We know this homeowners association wants to dredge through these seagrass beds. We don't want this to happen. We are your voters.
Yeah.
We don't want this to happen, and that's why I'm hoping that, you know, that the NEP will be a force for bringing together the citizens, the volunteer organizations. There's a variety of different volunteer organizations, and that as a scientist, I can help engage them, bring them information, get them, you know, get them engaged. It's not really my place to tell the policymakers what to do, because I think people have done that, and they're just not very receptive because, you know, if you know, they're worried about jobs, and I think it's having people who are voting that will have more of an impact.
Do you find the citizen scientists that you recruit tend to be more in the retirement age group, or
Yes.
are you getting a fairly good cross section of?
Definitely definitely more retired people, but young parents.
Okay.
Yeah, I do have some number of those.
Great.
Yeah, it's generally more retired, but not exclusively. Yeah.
Cool. Thanks.
Have a question?
Yeah, I had a question about the artificial reef search. You were saying that the longer that the limestone rocks were down there, the more developed the communities became, and then the more that you were seeing that nitrate being produced. So at what time point did you see that shift in the community? Like how long?
So I think I have to go back in Kendra's thesis. So the artificial reefs were deployed at the end of September, and I think the nitrate fluxes shifted and became positive. It was either her February or her April sampling period.
So it was within a few months though, it didn't take
Yeah.
years. Okay.
No, I mean her study was only a year.
Okay.
I mean it was yeah. Yes.
Because that's kind of interesting, because I work in aquaculture a lot, and we have tanks for bacteria that do that. And to cycle a tank to build a filter and get the bacteria to colonize it to adequate levels, usually, depending on what we do, is usually a month to 3 months, depending on the temperatures for the bacteria. Even from a totally sterile tank, we can. So it seems about right that, you know, there's this lag time for the different types of bacteria to start colonizing.
Yeah, definitely.
So one of your earlier findings was when you challenged a community that was living under hypoxic conditions with oxygen, you saw a more immediate response in nitrification in that community than one that had been primed with higher oxygen. Am I interpreting that?
Well, yeah, I don't know that. I guess what I was really surprised at by that experiment is that, you know, they came from this hypoxic condition, and you start the experiment, it only runs 24 hours, and so they it seemed like they were able to respond, you know, fairly quickly to that change in conditions.
Mhm.
So I'm not sure that the hypoxic ones were necessarily higher than the oxic ones.
But their response was faster to the change.
Well, I mean it was less of a change for the oxic ones. I mean, they were already, you know, the oxygen levels were already fairly high. So, yeah.
Okay. And I guess you answered this, the second part of that question was, I wanted to know the duration of that exposure, so.
Yeah, so we take the sediment cores and, you know, set up the experiment, you know, and then initial and
Mhm.
final measurements, so 24 hours between collection and you know, the final result. So not so long that you would likely have a completely different microbial community. I mean, obviously there are always some changes. You take something out of the environment, it's going to start changing, but
Yeah, I was curious what would happen if you allowed it to return to a hypoxic condition and then challenged it again with oxygen, because that's something you see a lot in toxicity studies, of community that's, you know, exposed to an adverse condition, and then you make the conditions good, and then they go back to adverse, and then you have a complete decline in your population. 'Cause of
Yeah,
this.
that'd be an interesting experiment to do that. So I've done, so I have done a variety of experiments where I've done different kinds of treatments. I've done oxygen and sulfide treatments, you know, for like a acute sort of 4-hour period, and then removed it,
Mhm.
and then oxidized everything and looked at the response. But I haven't then gone back and kind of done it again to see what they would do. Yeah,
Thanks.
that'd be kind of fun.
One more question? Oh, good, I have one. The huge drop in oxygen between June and July, and then it's low in July and August, and then it comes back up, and I realize there's a temperature thing. But it seems to me like it's pretty hot in June, it's pretty hot in September. So what causes that huge drop? I mean, if I remember the slide right, it went down from like maybe you know, 5 or 6 down to below 2.
Right. Yeah. So a lot of it can be, you know, what the conditions are in the river for stratification.
Yeah,
There
that's
it
the
is.
one.
Yeah. So yeah. So yeah, while September is still warm, I don't know if we you know, potentially we'll have fronts or hurricanes moving through, so we might have enough so we get some more amount of mixing. Yeah.
I mean, we see something very similar in Mississippi Sound over the, you know, off past Christiana, where the oyster reefs are. You know, we have numbers up in the 5, 6, 7 range routinely, and then you get down into July and August, and they usually don't get down to 2. Sometimes it's 3 or 4. There's a distinct drop in oxygen over those few months.
Yeah.
And, you know, temperature-wise, I mean, it's just as hot in September, so it's just a little odd to me.
Yeah, I'm not sure it's just as hot. I think, you know, the day length is getting shorter, and so I think you do see a little, you know, a little bit of an uptick. So you might get a little bit less, you know, decomposition.
Gene, is that over multiple years?
Yeah. So this is a long record. So this is like the 40-year record.
Mm-hmm.
So.
We've got, if you think about it, over 40 years, there's bound to be things like tropical systems that tend to overturn things around that time of year quite a bit, so.
Right,
That would
so,
bring the average up.
yeah, so, you know, so this paper, we were focused on this kind of more historic perspective, and so, you know, instead of looking at all of the individual year records, we were trying to composite it, so, you know, it's from one, you know, basically this mesohaline region. There's three sites that could have gone into the measurements as well, but so you can kind of see we hardly ever go out and sample in January. Right, hardly any January measurements, and September and October I routinely take my class out for a class that I teach, so that's why there's so many measurements in September and October. So, you know, again, kind of playing into, you know, if we were. So maybe the story from this is that I need a better analysis than a box plot. That this may be a little bit misleading in terms of, you know, the you know, kind of maybe doing a little bit more stratification of it in some ways. So yeah.
Well, thank you very much.